From Microbe TV, this is Immune, episode number 44, recorded on May 12th, 2021. I'm Vincent Racaniello, and you're listening to the podcast about the body's defenders against disease. Joining me today from North Carolina, Steph Langle.
Hey there. Good to be back. I'm glad that we were able to meet. I know we had to skip a month, crazy busy schedules, but I'm glad to see all your faces.
From Ithaca, New York, Cindy Leifer.
Good afternoon. How are you?
Looking very professional in your office there.
Sometimes I'm here, sometimes I'm not. Usually it's Thursday, Friday, but the AAI meeting is this week, American Association of Immunologists. And so I've been coming in and watching some of the talks here, so it's a little quieter, less disruption.
You have that post-tenure glow about you.
Well, it's post full professor.
But gotcha.
She was already tenured.
Since we're on video, my students made this for me.
Oh, that's nice.
So nice.
That's very
okay,
nice.
sorry. Post full professor glow. Thank you.
Good stuff. And from Madison, New Jersey, Brianne Barker.
Hi, it's good to see you guys. Good to be back.
Yes, Immune had a little hiatus, right? So we're back.
Okay.
If you like what we do here on Immune and all the other podcasts, consider supporting us. Microbe.tv/contribute. What are we talking about today, Steph?
Yeah, so the body's defenders against disease. And the disease that we're going to talk about today is a disease that I actually didn't look up the proportion of people, but it's multiple sclerosis, or MS. And this disease it's a demyelinating disease. I'm going to talk a little bit about that. But what it does is it causes these bouts of clinical and cognitive disability that go, it's kind of the influx. So you experience this and then you go into remission and then you have it peaks again and then you go back into remission. So this kind of like relapsing, remitting portion of the disease. And that coincides with inflammation in the white matter of the brain, spinal cord. So MS is really about inflammation in the brain. And what has been shown is that this inflammation is associated with the migration of lymphocytes and that they have breached something called the blood-brain barrier to then go on to cause pathology. And again, this demyelination. So your myelin sheath protects axons. And if you have inflammation or inflammatory chemicals and cells that are secreting chemicals that would disrupt or demyelinate the sheath, and then you end up having cognitive impairments. So this disease there are current treatments. What's interesting is that even though over the course of, so somebody who has MS, and we'll talk a little bit about how that's diagnosed, but somebody that has MS, they have this relapsing remitting, kind of these peaks of disease that kind of go back down. That relapsing remitting cycle will slow down over time, but the atrophy of the gray and white matter in the brain continues to increase. And so they don't really know why that happens, why you just go into this chronic phase. So the two phases are relapsing remitting, and then the second is this chronic. As you get older, you tend to continue to have atrophy of the brain and cognitive impairments, and then could end up very low quality of life, essentially. So with this disease,
Can I ask you something? There are some individuals as well that have a progressing form, right? So I think there's two forms of the disease.
Yes.
And it's interesting because I'm sure you're going to get into this, that there's mouse models.
Mm-hmm.
And most of the mouse models model the progressive form of the disease rather than the relapsing remitting form.
Yes.
And so I think it's important to keep that in mind when we're talking about molecular and immunological mechanisms of disease. When we're thinking about the different forms of the disease.
Yeah, it's a good point. So you're right. There's a proportion of people that would not have the relapsing remitting. They would just progress to having the chronic form.
Exactly. And for people who are not up on their neurobiology, the sort of connection here between the myelin sheath and how you end up with some of the neurologic issues that we have is that the myelin sheath is basically sort of a lipid layer around the axon that helps make sure the electrical signal transmits in the sort of right direction. I kind of think of it like the plastic outside a wire that makes sure that the electrical signal is going in the right direction and not sort of skipping around through the axon. And so it's really kind of the same idea of if you have a wire where the plastic on the outside is frayed, that you're going to have shorts in your electrical circuit. And the same thing, once you actually are losing some patches of that myelin, your neurons are not going to be able to conduct as well.
And it's mostly the motor neurons, right? So that you get this progressive paralysis. But there's also the other kind of neurons that carry information as well and store memory and things because they have cognitive dysfunction as well, right?
Yes, they do.
Right. Right. And so you can imagine it's a pretty, depending on the form you have, the treatment which you're going to use, you may not be responsive. And we're going to talk a little bit about the different types of treatments that have been effective. But the main players in this disease have thought to be T cells. And that comes from animal models, as Cindy mentioned, where you could prime a T cell with the myelin protein and have so these T cells in the mice, if you transfer them to a naive animal, those T cells are sufficient to induce what is called, the multiple sclerosis equivalent in a mouse, which is experimental autoimmune encephalitis, or EAE. And that is a very common model, although it doesn't recapitulate all aspects of this disease. So we mentioned that this disease has multiple different forms, depending if it's more acute or chronic. And so you can prime the animal. You can have this like active induction of EAE, or there's a viral induction. You can use I forget the virus they use, that kind of helps to model the inflammatory process, but then it may not model the chronic as well as the EAE. The EAE, I think, is a very reproducible model because you're using the same protein to be able to induce this. And so when you're choosing your animal models, you just have to consider what are you trying to model. But I think in the field, and Brianne and Cindy can let me know, I mostly see EAE. I don't often see others. I'm assuming the reproducibility, right, is why.
Yeah, and I know that there's a two-step thing that you have to do when you're setting up this model. And the first thing is you have to immunize with the peptide, but then you have to break open the blood-brain barrier, and they use like a cholera toxin or something. I forgot what it's called. The toxin that you add in it allows the T cells to get into the brain.
Right, and so that goes back to thinking about how MS is caused, that it can't just be the peptide alone. The body's not going to recognize it. It's a self-protein, but it's something about inflammation, breaking the barrier. So pertussis toxin, cholera toxin, Freud's adjuvant, I think they use also in conjunction. But you're right, it is a two-step process. You inject the protein and then you inject essentially the adjuvant.
You basically have to break tolerance because this shouldn't happen, right? Because you should be able to harbor self-reactive T cells in your body, and they should be controlled under normal circumstances by peripheral tolerance. But somehow in this model, peripheral tolerance is broken, and those cells then go and attack the myelin sheaths because they express, parts of the myelin protein, myelin oligodendrocyte. MOG.
MOG is
MOG
is myelin oligodendrocyte glycoprotein. It can also be MBP, myelin basic protein, which I learned from one of my colleagues in grad school is named that because it is a protein that is not an acid, not because it is basically what myelin is made of.
I love that.
I love that. Good. It's good to know. It's very informative, that name.
And the 30,000 foot view of this is if we understood the immunological and molecular mechanisms that drove the disease, we could then try and interrupt those processes. And I think that that's the goal of this. And so as you're going to talk about, I don't think you talked about the title of the paper yet.
No, I will. Yes, I.
But if we understood these molecular mechanisms, then we could begin to target them. And this paper that we're talking about makes obvious some candidates that one could use to target the disease.
Yeah. So actually, before I talk about some of the treatments that are used for MS, because this was used the authors used the information that they got from clinical trial, that others had gotten from clinical trials to inform their hypotheses in this paper. So this is a Cell paper. It's called Recirculating Intestinal IgA Producing Cells Regulate Neuroinflammation via IL-10. And the first author is Olga Rojas, and the last author is Jennifer Gomerman. And this group is from the University of Toronto. And they have been doing a lot of work in IgA biology and trying to understand MS using multiple different mouse models, which we'll get to talk about today. Genetic ways of asking the same question, but to get at an answer. So anything more about the paper?
There are 3 lead authors.
Oh,
we don't want to leave them out.
Don't. See,
and now I'm going to have to try and pronounce it.
Thank you. I almost forgot.
To do that. But yeah, there are 3 lead authors.
There are 3 lead authors.
Olga Rojas, there's Anne-Catherine
Probstil, and Elisa Portafillo.
No, there's no T. Portfolio.
Portfolio.
There's no T. Portfolio.
No, there's no T. I wanted to say T.
You guys want to say a T in there?
I know we do.
Okay, we're terrible at pronouncing names, but I just didn't want to leave them hanging.
Yeah, thank you.
Because a Cell paper this large has a huge team of people that need to produce.
Big team. I mean, we don't
want to leave out the first authors.
Right. There are patient samples, MS patient samples that we're going to talk about that could have been like a JCI paper alone. I mean, that one little section. So these papers are quite dense, and there's a lot of co-authors, but you can look up that paper if you want to see all the different co-authors from multiple different schools and a lot of collaborations that went on.
Yes. In fact, it's an open access paper, so you can take a look at it.
Go forth.
Have access.
Great. So there are some drugs that have been shown to be effective. So one of them is rituximab. We have talked about rituximab before. This is it treats B-cell lymphoma. So it's an anti-CD20 antibody. CD20 is a receptor marker on B cells. But what we will learn is that it is not a receptor on all B cells. So to go a little bit into B cell biology, B cells are coming from the bone marrow. They come as stem cells, and they go through this developmental stage where there's B cells and pro B cells, and they transition out of the bone marrow niche into circulation, where they then go to different tissues depending on the stimulation that they encounter. But in particular, when they differentiate into antibody producing cells, which is the effector cell for B cells, right? That's actually going to be producing the thing that we care about, the antibody that's going to block whatever's going to happen. It loses expression of CD20. So you'll find CD20 expression through all of those stages until it gets to something called a plasmablast, which then differentiates into a plasma cell. So in this paper, a lot, they talk about PBs and PCs, plasmablasts and plasma cells. They're terminally differentiating. Plasmablasts are a little bit, I'm going to say, younger than plasma cells. They're in that differentiation state. They're transitioning from the B cell that has gotten the signal in the germinal center to undergo classic recombination and somatic hypermutation to then become this mature plasma cell. So rituximab, if it blocks anti-CD20, we can imagine that it is not, in fact, blocking plasma cells. And so what was interesting is that anti-CD20 was effective at treating MS. So the burden of lesions goes down to a burden of new lesions essentially would go down to 0. And I think it's known as one of the best treatments. But there, of course, was a group that was resistant to anti-CD20 treatments. And in those patients, they had explored different types of drugs to try to block B cells. And so what's interesting, right, is that we think about this as a T cell mediated disease, but the fact that rituximab works suggests that B cells play a role, that it's not just T cells. So if, anti-CD20 is working, but in these patients that were resistant, they tried something else. There's this it's a fusion protein. It's called TACI-Ig. The drug name is atacicept. Thank you. And what atacicept does is essentially blocks binding of essential factors that are important in B cell and plasma cell survival. And people who underwent this treatment actually did far worse. They had to stop the trial because people's MS clinical symptoms and pathology worsened. And even individuals who did not have MS but were predisposed to MS for, I think they had some other autoimmune issues, they actually progressed.
Yeah, and that is really surprising because the whole point would be if the rituximab was working because you're depleting the B cells and they were causing the problem making the antibodies, one
Right.
would think if you blocked all the survival factors, you would be better off. But this would suggest that somehow at least a portion of these B cells were protective, which doesn't make a lot of sense if when they're producing antibodies, the antibodies would be recognizing potentially the myelin sheaths or something else in the brain that's causing the inflammation. So how can this be working? So it was really
Totally.
perplexing.
Yeah, very perplexing, but super interesting. And from a B cell biology perspective, it kind of started to paint a picture maybe of what was happening, and that it may not be in fact the antibodies that are causing the problems, but secretory factors from the B cells themselves. Let's say a non-plasmablast or plasma cell B cells, so these kind of like more immature cells are secreting factors that could potentiate disease. But the plasma cells, there might be something about them because when you block them, the disease actually does get worse in patients. and a couple other just factoids, you can find B cells in the brain of these patients, so they are there. And I think in terms of introduction, I want to make sure I covered everything. But yes, so then one of the questions was, well, what is it about plasma cells? And the authors wanted to know if they could find them in the CNS during their experimental autoimmune encephalitis in their mice. And so the start of the paper is really just to identify those cells, and they use some pretty cool genetic models to be able to do this. So you can use the genes that are important in plasma cell development. And one of those genes is called BLIMP1. Well, actually, that's the protein. The gene is called PRDM1, and it produces the protein BLIMP1, which is important in plasma cell development. So if you hook a fluorescent protein into the sequence, and if that gene is expressed, then you can visualize it through flow cytometry.
Hey,
And so-
a factoid, my colleague,
Yeah.
Catherine Kalame, discovered BLIMP1. Did you know her?
Really?
Any of you know her?
I don't. Cindy, do
Not
you?
well. Mm-mm.
You heard of her though, right?
I've
Yeah,
never,
yeah,
yes.
but yes.
Yeah, she used
That's
to be
very cool.
in the department. She's retired, but
Is
that was her
she
thing.
retired?
BLIMP1,
Okay.
yeah. Transcription factor, I believe, right?
It's very
Yes,
important.
it is.
It is.
Yes, very important. So not only that was also used, but then there was another model where they deleted the enzyme that causes somatic hypermutation. It causes mutations in the DNA and RNA, and to help basically the B cell evolve when it's undergoing stimulation, presentation of an antigen. And so by deleting that enzyme, cells also cannot mature to their fully functional state producing antibodies. So by using some of these genetic tools, then they could see that during the chronic phase of autoimmune encephalitis in these mice, that you could detect plasma cells in the brain. One of the first questions they had was, are IgA producing cells detected in the CNS during EAE? So they focused on the chronic stage. So I believe in this mouse model, that is the stage. I don't think there's a relapsing remitting form in EAE. They really just have kind of this progressing to the chronic stage as observed. They in the supplemental, they talk about like how do they observe that in mice? I think there's like the foot pad or the leg, whether it has paralysis or not.
Yeah.
Yes.
Right. That's
The
kinda,
first
there's
thing that
stages,
goes is the tail. Their
the
tail just kind of drags.
tonality. Right.
Yeah, and then it's the leg, the back legs, and then it goes to the forelimbs. And then they used to be able to take it to, I think, a score of 4 or a 5, but they can't do that anymore. Basically, by the time they reach a 3, they're starting to get forelimb paralysis and they can't eat, so they have to euthanize them.
That's right. And it's interesting, even in the supplemental, they talk about they use a 16-point scale system to be able to more than, I think the typical is 6. So very robust, very robust. So what one of the things about plasma cells is there's no definitive marker. It can be very challenging to detect them because they don't express like one marker, CD20 or CD19. So they talk about in the paper, they're really defining what these cells express. And so we talk about low expression of B220, K67, and CD138. And so that's how they characterized these cells. And they observed a significant increase in the number of cells expressing kind of like this set of characteristics, what they're defining as plasma cells in the brain and the spinal cord at the peak of EAE, and then an even greater increase during the chronic phase. And so the graph they're showing is kind of like this increase in cells in the brain and spinal cord with the increase in disease. And so they wanted to characterize, well, what's the isotype? And so they did two things. They did intracellular flow cytometry staining for both IgA and IgE. So there are multiple different isotypes. I don't think we talked about IgM in this paper, but IgA and IgG are kind of the ones of main interest. And then ELISpot, which is a really interesting assay where you're basically detecting antibody secreting cells by coating the plate with your, let's say, either an antigen or just an IgA antibody. And then if the antigen, if the cells are secreting antibody, it'll bind to the bottom of the plate and you can see these little spots. I did a lot of these in my PhD. And so you count the spots and then you can detect like one spot would be a single cell that you're detecting in this assay. But of course, the challenge with ELISpots is that it's hard to distinguish between plasma cells and memory cells. There's no real way to distinguish those. So they do talk about that as kind of like a caveat. Now, what was interesting is they did detect both IgA and IgG. There's a bit of a flip. Using intracellular flow, they detected more IgA, but in ELISpot, they detected more IgG. And so I thought it was interesting that they went forward with the IgA question because there are IgG antibody secreting cells there.
Do we know that, for example, if you took a generic IgG producer and a generic IgA producer, whether they produce similar amounts of antibody on a per cell basis, or is it easier for a cell to make IgG? I don't know this because that could be one of the answers, because there are more IgA producers, but equivalent levels or high levels of IgG2, but maybe the cells that make IgG just make more IgG, and that's why you can detect more. Whereas, I don't know.
Do you think it could also be related, and I'd have to look at their methods on this, to the number of different subtypes there are? Because there are more different types of IgG. And so if you were to sum them together, they might come up to more, as opposed to the fact that there are only 2 IgAs, but there are 5 or 6 IgGs?
Yeah, that could be it. It is interesting. So the quantity of antibodies in serum, IgG dominates, but we actually find a lot in terms of just number of cells, more IgA antibody secreting cells in blood than IgG. So I do think it must be what you said, Cindy.
Yeah, that's interesting.
They produce IgG at a greater clip.
I had wondered that, but I didn't know that for sure. That's really interesting.
Yeah. So they wanted to move forward with the IgA to ask, like, how is IgA getting in the brain? That's not particularly a cell type that you think of being in the brain, because the majority of IgA antibody secreting cells are coming. They're in the mucosa. They're coming from the gut. The gut is this really robust organ that is chock full of cytokines and stimulating factors that would potentiate B cells to isotype switch to IgA. And so we actually think of a lot of circulating IgA cells in the body to be coming from the gut.
Yeah. So Steph, I kind of have a question for you about how you feel about this. How
do I feel about this?
Well, so I usually, we often think about IgA as being part of the gut, and we think about mucosal immune responses. And targeting IgA types of responses in the mucosal tissues and things like that. So here we're seeing this importance for IgA outside the mucosal tissue. In fact, I was really interested when you just mentioned the IgA you're seeing in the blood. And so as someone who's spent some time thinking about mucosal tissues, I wondered how did you find this very surprising, or did this seem to kind of make sense for you?
Well, I mean, I don't think about the brain a whole lot, but we're learning a lot more about the gut-brain axis. So 2 things. One, yes, I would expect there to be IgA secreting cells there, just because everywhere I look, I find them. They're in the mammary gland. I find them in the NALT, which is the nasal lymphoid, actually a little bit less in the lung. And it depends on kind of and we're going to talk about this, the inflammatory state of the animal. Have they been infected with a virus? Are they undergoing, an autoimmune disease? So I guess I was surprised because it's the brain, and I just didn't the blood-brain barrier, I thought, is always, pretty tight and wouldn't, but I guess during a disease state, there they are. If they're in blood, they're going to go.
I think when you have inflammation, it reduces the
BBB.
intactness of the BBB. Yeah.
Yes.
Yeah.
So
one of the things that I don't think I saw, I didn't look at every supplementary data figure. Do they talk at all about dimeric IgA versus monomeric IgA? Because my presumption is that your experiments where you see IgA in the blood is going to be monomeric IgA. That's what I learned when I learned immunology, and that the dimeric IgA is produced at the mucosal surface because you have the poly-IgA receptor that will transfer the dimeric IgA to the lumen, and it's stabilized so that it can bind to microbes and whatever else in the lumen.
Yeah.
But what would be going on in the brain? Do we know?
I know. So that would be one question that I would like to email this team about is, did they do, I didn't see it in the paper, but an anti-secretory component or anti-J chain antibody? Does the brain express, because PIGR is the receptor, and does it express that? So in blood, you mostly expect to see monomeric, but if it's coming from the gut, it would be, they would be secreting dimeric IgA antibodies. So I think that's a super interesting question, because you do have these two different types of antibody secreting cells depending on where they're stimulated, I guess.
And it would change the functionality of the IgA, wouldn't it?
It would. And a little bit later, one of the animal models where they induced this B cell activating factor at high levels, at transgenic mouse, the type of IgA antibody is dependent on the glycosylation of the IgA and can play a role in nephropathy and kidney disease. And so, I think the roles of IgA are dependent on there's dimeric or monomeric, how it's glycosylated. And we don't really learn that in this paper, but I'm sure maybe that's going to be their future,
Yeah. So I guess the big thing is, do you believe it? Do you believe their data and the fact that the premise of this paper is that there are in fact detectable IgA producing B cells in the brain of these EAE mice? Yeah.
I definitely buy they use multiple different animal models, genetic mouse models, to try to understand this. And even as reading it, the ELISpot isn't always as, for a reviewer, I think they wanted additional, secondary markers. They use some pretty cool thing called CADE mice, which are like fate mapping mice where you can actually see the cells by fluorescence. So I do believe their data. And we'll walk through a couple of the animal models to show that they did see these IgA antibody secreting cells in the brain, and that maybe they're protective. I would think that antibodies ending up in the brain might not be such a great thing, especially in an autoimmune disease. But seems to be the case.
But at the same time, one of the other big treatments for MS, at least in the past, was Tysabri, which was meant to block T cells going to the brain. To stop T cell trafficking to the brain. And when the patients got Tysabri, some of them ended up with PML because they had reactivation of some viruses that were in the brain. And so, and in
viruses,
fact,
pesky viruses.
And so the T cells couldn't do anything there. And so maybe we do want some antibodies making it to the brain just to help out with surveillance.
For sure. Yeah, absolutely. So the next big question is, do these antibody secreting cells impact the clinical course of the disease in their mouse model? And so, yes, in fact, they did see that in plasmablast and plasma cell deficient mice. So they had genetic mice that they did not have these cells. They did exhibit exacerbated EAE and higher mortality compared to littermate control. So then that's the question that kind of kicks off this paper, as like, okay, they're in the brain. And when you remove them, the disease is worse. So after that, they asked the question, well, where do they come from? I talked a little bit about how that, the majority of IgA antibody secreting cells that we see migrating do come from the guts. And, they also have observed that in exacerbated EAE, that there's a decrease in the number of plasmablasts and plasma cells in the small intestine. So they call this SILP. I kind of sometimes, when I was reading it, it looked like SLIP, but it's small intestinal lamina propria, which is really the tissue space below the single layer of epithelial cells in the gut, all that lamina propria. It's really meaty with lots of cells and lots of chemotactic factors. And so the thought is, if you have a decrease in those cells in the gut, they probably are migrating out of the gut and going other places, potentially the brain. So to test this hypothesis, whether gut-derived plasma cells can suppress neuroinflammation, they did a adaptive transfer study. So they pulled out cells from the SILP, the small intestinal lamina propria, that were either, let's just say, non-plasma cells or plasma cells. And they define that by different markers. So B cells that are not differentiated into this plasma cell state or the plasma cells. And then when they looked at these mice, only the ones that were transferred with the gut-derived plasma cells. They were able to basically have no effect on EAE. So the ones that were transferred with the non-plasma cells had worse disease. And they did this in a couple different other genetic mouse models to really prove that point. And then they could also find these cells after adaptive transfer. Not only did they protect the mice against the disease, but they could find it in the brain, in the bone marrow, and in the lymph node of the recipient mice. So they're reaching these tissues. Confirming that the at least for gut derived, they didn't really do any other adaptive transfer studies to see like other mucosal sites, can they act this way? The focus of the gut was obviously because those cells, there's just a huge amount of them that come from the gut and they do protect. Super interesting.
Yeah. And these,
so they kind
these
of go
IgA
into this
were all different specificities, I suppose, not monoclonal or anything like that, right?
Yes. So that is a weird thing, right? Because why would you just willy-nilly recruit B cells with polyclonal specificity, right? Like just many different types of B cells. It also suggests, when we started talking about this, is there a role for the antibody in this process, or might it actually be something else that's antibody independent being done, right? Because then it wouldn't matter what your specificity is.
You could do that experiment.
So maybe just a B cell who switched to IgA also has some other function that happens to correlate to having made that switch.
Yeah. And we can even hop, because it's really interesting. They go to like the middle section of this paper. They really explore like where do these gut IgA antibody secreting cells go? And they use infection experiments with two viruses that we are familiar with on this show, rotavirus and influenza. And then they kind of, after that, they go into the further like molecular mechanisms of what you just said. Is it the antibody that's doing, or is it a factor from the cells that are decreasing the symptoms of EAE? So I think what I'll do actually is I will hop back to those studies of like, where do these antibody secreting cells go? Because I think it's super interesting to talk about trafficking. But let's skip ahead to where they talk about more mechanistically, like, is it the antibody? Mm-hmm. And so they were able to use mice that did not express IgA. So the cells still survived, but they did not express the antibody. And when they did not express the antibody, it didn't really matter. So the severity of disease was still the same as the wild type. So suggesting that it's not the antibody that matters, it's something secreting from the cells themselves. And that kind of sent them on this trajectory of asking the question of, what is being secreted? And there has been a lot of recent work that these plasma cells secrete immunoregulatory proteins like IL-10 and IL-35, that their focus is really on IL-10. And we've talked about IL-10 in the past, but it's a anti-inflammatory protein. It really helps to suppress inflammation. And if MS is really this, defined as an inflammatory disease, and it's not the antibodies that are, if the IgA double negative mice have the same severity of disease, then it's something secreted. And so what they did was they took a couple different approaches to knock out IL-10. And so when you knocked out IL-10, you had a greater disease severity in these animals, suggesting that intrinsic IL-10 secretion was required for suppression of EAE in these animal models. And they used, I've never used these, but they're called 10-bit. Is that how you pronounce that? these 10-bit mice. Essentially, if the IL-10 gene is expressed, there's a particular surface protein that's expressed that you can pull out, and then you're only then pulling out cells that express IL-10, and you can look at what those do via ELISpot, which they looked at, mRNA and things like that. So they, I think in that experiment, at least proved to us that IL-10 is one of the regulatory factors that help to suppress EAE in their mice. They also looked at NOS2, which is the gene that encodes INOS, nitric oxide synthase, correct?
Yes.
Yes, nitric oxide synthase.
Inducible nitric
Inducible,
oxide synthase.
Right. And it's secreted from monocytes, myeloid cells, to be able to, it's essentially supposed to kill infected cells, but it ends up causing a lot of damage to the tissue. And so when they knocked out NOS2, they did see a mild phenotype, but it wasn't as strong as when you knocked out IL-10. So the disease was exacerbated, but not as strong. So suggesting that IL-10 really is the key factor here. And INOS may be, it to a lesser extent, also playing a role. So I think I was convinced by the IL-10 data. What do you guys think?
Yeah, I think so too.
Yeah, no, I think so too.
Yeah. They went on to look at some different transgenic mouse models where they overexpress, I had mentioned before, this a B cell factor that it helps to differentiate into plasma cells. It's called BAFF. And by overexpressing BAFF, they saw an expanded pool of IgA plasma cells in the small intestine, which I guess, would make sense because you have basically overproduction of a cytokine factor that helps B cell maturation. And so what was interesting in these BAFF mice is they actually they made a big point in this paper to distinguish that at baseline in their typical models, they don't see antibody secreting cells in the brain. It's only during EAE they see that. But in these BAFF mice, they find IgA antibody secreting cells in the brain at steady state. And it was not further elevated during EAE. If you then immunize the BAFF mice with the MOG, the myelin protein, they don't develop clinical symptoms at all. And so it suggested that by overexpressing BAFF, you have an increase in IgA antibody secreting cells in the brain, acting as a protective factor before the disease onset even occurs. At least that's how I interpreted it. So they do not develop clinical symptoms at all. And they tried to recapitulate this by using a recombinant human version. And it really, likewise, they were resistant to the induced disease, potentially because of the surplus of these cells in the brain.
And that's really interesting because one thing we really don't understand with MS or many different autoimmune diseases is the initiation events.
Right.
And so the fact that this can actually stop initiation of the disease, as well as kind of the propagation or the disease sort of staying around, is really interesting because this at least gives us some ideas of things that are important in an initiation phase.
Right.
One would wonder then if there are potential genetic polymorphisms in these various genes that might change expression levels or steady state levels of these IgA producing cells in the brain that might predispose these individuals. I don't know.
Right. And one of the things we don't really get to dive into is, how are they trafficking? I think they talked a little bit about potential chemokines that help to the cells have to get there somehow. And so differential expression of those chemokines and, the trafficking cues maybe could also play a role. And they do some other really cool transfer experiments where they take pre-primed T cells from donor mice immunized with the MOG peptide that resulted in ENE. And then when they transfer them into wild type recipients, the transfer of those same T cells into the BAFF mice resulted in significantly attenuated EAE. So further, there's something about these BAFF mice and their ability to increase IgA antibody secreting cells. They also wanted to ask, what about the T cells? Like is there, they didn't really dive into that too much, but they found a reduction in the frequency of CD4 T cells that express GM-CSF. There are some key cytokines in this disease, one interferon gamma, IL-17, GM-CSF, which is granulocyte. What is it, Cindy?
Monocyte
monocyte
colony stimulating factor.
Yes, thank you. And so maybe, further suggesting that it is the interaction of the things being secreted by the B cells that stimulate other cells like T cells to produce GM-CSF, and it's like a pro-inflammatory cytokine.
Yeah, it would have been nice. I mean, they did so much in this paper. It's hard to ask for more, but it was a somewhat myopic and biased way of looking at this. They had a culprit and they looked at that culprit and they saw a difference, and so that must be what's going on. However, as you say, like the T cells probably are playing a role as well. And
yes.
so
like what are they up to?
You're looking in a more unbiased way as overall what is happening in these, for example, the BAF overexpressors that might be different because it may be BAF or it may be another in the B cell, or it may be another cell that is able to also participate in this phenotype. Just because there's more B cells doesn't mean that they're actually the ones. I mean, there's a lot of circumstantial evidence, but it's still also somewhat, we looked for it and we saw it, so that must be what it is.
Mm-hmm. Yes. And I think in regards to, this model, so the BAF transgenic mice, they're overexpressing a protein. I mean, that's not really, you're not typically overexpressing BAF. But from what I know about this model, they do have an increased amount of other autoimmune diseases. So I'm actually really surprised that in EAE, they don't have there's not this issue with EAE. And I guess because if the phenotype is truly, if you have more IgA plasma cells, then you have reduction in EAE. Whereas for other things, and so I'm particularly thinking about diseases that affect, nephropathies where IgA is getting stuck in the kidney, and they're stuck there because they are misglycosylated and they kind of aggregate together because they're not properly formed. They experience that type of disease with these BAF mice. So I think it's kind of like, you overexpress any protein and you're bound to have like other issues. So I would be interested in, like you said, Cindy, what are they, dimeric, monomeric? What's their glycan structure? Things like that.
Well, and that also actually ties into the trafficking question. Because one piece to all of this is, we have these plasma cells. We're realizing that these plasma cells are making IL-10. They're not just antibody producers and only antibody producers. They have this important cytokine role.
Yeah, sure.
But is it important that they are specifically going to the brain, or would they go to a site of inflammation elsewhere?
Right.
And so is this a specific gut brain thing? Or if there were inflammation in places other than the brain, would we be able to have these same IL-10 producing plasma cells reduce inflammation? And given that, as you say, the BAF overexpressor mice have other autoimmune diseases, that might give us a hint, although obviously there are a lot more experiments that you could do.
Of course. Yeah, definitely. And so what I'm going to do now is hop back more to the middle of the paper, because it's really interesting, your question of about trafficking and other places where there's inflammation. And I think it matters where the cells were stimulated first. There's just one last experiment they did. So I talked about BAF. BAF is a secreted stimulating factor for B cells. So is APRIL. They're highly expressed to help produce these plasma cells, and their ligand is TACI. And TACI is a TNF family receptor. And so when they had these knockout mice, they also showed evidence of exacerbated EAE. So it's just another way to show do you think that helped explain those initial patient studies where when they used the TACI-Ig, they had worse disease? Right. Yep. I would. Yep. Because that was the fusion protein that blocked that interaction of the BAF or APRIL binding to TACI. So you can't develop plasma cells because you don't have the signals. Right. So I really did like kind of the trajectory of understanding, okay, the IgA, it's in the brain. Taking it away exacerbates disease. It's not the antibodies, because when you have a plasma cell that can't secrete IgA, it doesn't matter. So it really is the IL-10 that's being secreted from the cells. But I do want to go back because they really dive into some really interesting findings about commensals. And so I had somebody ask me a question the other day of, what proportion of IgA in the gut is against commensals and what proportion is against pathogenic species like viruses. And I couldn't answer that,
lot, right?
but it's a lot. It's going to be a huge proportion. I mean, we secrete 3 to 5 grams of IgA in the gut a day, which is a crazy amount.
A lot.
A ton. And they're not necessarily binding strongly to the commensals. I think they're more there to kind of anchor them and also to make sure they don't penetrate the gut. You're trying to keep them under control, right? You're trying to maintain a homeostasis. So IgA against gut commensals. Oh, actually, maybe should I go back to the rotavirus influenza story?
I think you should. We skipped over that.
Yeah, sorry about that. So before we go to commensals, let's talk about where else do they go? And answering Brianne's question of, is it just the brain and does there have to be inflammation? So what they did was they used rotavirus, and rotavirus infects intestinal epithelial cells, and you can get this robust response, IgA response in the gut. So you have really by day 12, a peak of IgA antibody secreting cells that have come out of the gut, and they actually migrate to the periphery, to the systemic. But they also used influenza because they wanted to understand, if you stimulate something in the gut, can you see these cells in the lung? Do you also see influenza-specific cells in the lung? And if you do both, rotavirus, wait for the rotavirus to clear and then infect with influenza, what do you see? So what they found was IgA antibody secreting cells were detected in the lungs, but only if rotavirus was involved, which is very interesting. It kind of tells us that the lung is not a good inducer of IgA antibody secreting cells, that you need the stimulation from the gut and they migrate out into periphery into the lung. Because only in the mice where they had either rotavirus alone or rotavirus plus flu, as early as like 2 days post-infection, you see IgA in the lung.
I wonder, so they're looking at the lung here. I don't actually know if you could do this in mice. This might be a really bad idea for an experiment in mice. But I wonder if you could, when we think about some of the SARS-CoV-2 literature, we've been thinking about IgA in the deeper lung versus in the nose. Could they have looked in the nose of the mice?
Oh, so that would be I would love that experiment. Yes. So there is seemingly a stronger axis, if you will, from the gut to the nose, to the nault. I think it's because there are higher levels of things like CCL28 and CCL25, which are kind of these key IgA regulatory ligands that help to bring the cells to the site. The lung is weird.
Yes, I think that's what I've learned about lung immunology is summarized by that.
It's very weird. Their lymphoid tissue is inducible. You don't really even get a lot of cells to begin with. So I don't know why it's not a good producer of IgA antibody secreting cells other than, and Brianne may know because I know your sister studies this, but there's not a lot of lymphoid structures, correct?
Yeah, I think that's more inducible lymphoid structures.
Right.
Yeah.
Right.
Yeah. I'm curious though. Okay, so the idea here is you ingest, let's just make this. You ingest rotavirus, you get rotavirus infection. You now have rotavirus specific IgA producing B cells in your gut. Now you get influenza in the lung, and what these experiments are saying is, I guess, just in case your gut specific that are specific to rotavirus now go to wherever the flu was. Why evolutionarily would we do this? Would you expect co-infection to happen frequently, or do we think it's this regulatory activity that's important, or why would this happen? Yeah.
So basically is the idea, I guess the way I read it, and maybe I think I was making assumptions here, was that these are really good IL-10 producing cells and we send them wherever the inflammation is happening.
Yeah, I think that right. It's more this circumstance that I believe that cells that are, they're stimulated in the gut, a lot of them stay in the gut. There are, things like alpha 4 beta 7 that help retain them in the gut, but there's a proportion of them that just, they circulate. And I think it's just, at the time they're circulating, the lung starts secreting chemotactic chemicals, and they are in and around the lung. A small proportion than what we see here are going to end up there. So I don't know if it's a evolutionary thing or it's just by circumstance, because like you said, it's just better to have these cells circulating at all times because they're secreting protective factors, even if it's, against the wrong pathogen. Like this is a monoclonal, the B cells to rota, but it's still secreting things like IL-10.
Yeah, I mean, evolutionarily, that would make sense because it would just generally help to dampen immunopathology.
Right.
So that's what you're thinking IL-10 is doing. If it goes to the lung, it's going to dampen immunopathology, right?
Yeah.
That's what you're thinking? So I wonder if there's a correlation in people between, rotavirus and influenza and MS.
Right. Well, I mean, I think influenza would be a good pathogen to look at just because rota as in adults is challenging. We don't really get, we're stimulated by rotavirus ever since we were kids. I guess with influenza, we are too.
Yeah,
I don't know. I'm just trying to think of how that would work. Yeah.
I would play devil's advocate. Why is it important to bring this B cell from the gut make IL-10 when we've got other cells whose job it is to make IL-10 and regulate inflammatory responses in various different tissues?
Yeah.
Like, why not call in a T regulatory cell or even a B regulatory cell? They exist, right? So why these? And so my thought is that there must be a reason, because they are also making antibodies, and that you would want them to be there. I understand that for the regulation in the EAE model, the antibodies are dispensable. But for something like this, when you're doing an infection with one virus and then an infection with another virus, it may be that you would want those virus antibody producing cells to come there in case you got exposure at that mucosal site. Because there's this idea of this reticuloendothelial system where all of the, the common mucosal immune system is kind of old nomenclature at this point, and it's sort of falling apart, especially with now we have this gut-brain axis thing. But the idea is that, if you make immune cells in one mucosal surface, they can get to other mucosal surfaces.
Right. No, I would agree that, because even rotavirus has a brief systemic period. So having antibody secreting cells that are in circulation when rotavirus has this brief viremia would be beneficial.
Yeah.
And could you have this, could you have rota in the lung, even though it's not going to replicate there?
Yeah.
You could still, it is. I think depending on the tissue, it could just add that extra benefit of protection in case.
If you're an immune cell making an antibody, you don't necessarily know that the virus is able or not able to replicate in the tissue in which it's in, right? We know that because we've studied that. But, if you're just making an antibody to a pathogen that is a problem, then it might be you just go there just in case as a reserve troop or something. I don't know.
Steph, did you say that IgA responses in the lung are poor compared to the gut? Is that right? But not the upper tract.
The nose is good.
The nose is good.
Yeah, and so there's I forget there's a really nice experiment done, I think in the 90s, where they were giving, I think they gave recombinant antibodies, and they saw in the blood, and they were able, the IgA really trafficked to the nose, and the IgG really trafficked to the lung. And so why that is definitely the chemokines they express, but then the ultimate why, I don't. But so I think I go back to SARS-CoV-2. If you can stop a respiratory infection from seeding itself in the lung at the nose, and IgA is the only antibody that's super good at getting itself into mucosa and has that protease resistant secretory component. If you can block things up here before they get to the lung, where things go systemic more quickly, where IgG may matter more. This is kind of what I'm thinking about.
Yeah. Well, and part of it also, when I think about that, is just that if you make a good IgG response to, say, a respiratory viral infection, that might protect you from disease, but may not protect you from infection. And it may take the IgA in the nose to be sterilizing to protect from infection.
I'm also thinking that most respiratory viruses are swallowed at some point, right? Because the mucus is coming up. And so, even though they don't reproduce in the gut, you're getting a lot of antigen, and maybe that
Yeah.
is producing IgA. That thing goes back to the lung. Maybe it's a built-in system to do that. The mucosal elevator does two things, right?
Yeah, for sure. Yeah, I like that thought too, right? And we talk about respiratory viral shedding in the gut, and even if it's not replicating much, it's still there. So, yeah. So it was very cool. I appreciated, using two different viruses to look at two different organ tropisms and try to understand trafficking. They also did these parabiosis studies where they surgically attach mice to each other. And then they have one mouse that was infected with rotavirus. They let it clear. They attach them, and then they try to see in the uninfected mouse if those cells move from the one mouse to the other. And they do find those cells in the uninfected mouse, of course, at lower levels, but they were able to detect them. They use these CADE transgenic. Essentially, if you shine violet light over, you can photoconvert these cells that express CADE red, and they turn from green to red or red to green. I forget. Green to red.
I think.
Yeah, green to red. And so I think they did this. It's just another way to detect rare cells, because if they're at low levels and you chop up the intestine, you try to run ELISpots or maybe flow, this is just like a third way to try to find these cells, would be my guess. They also asked the question, in these rotavirus infected mice, they wanted to relate it back to EAE. So they examined the CNS for presence of the rotavirus specific antibody secreting cells. After the animals cleared the rota, and they confirmed that rota doesn't impact the severity of EAE. These cells are absent in the steady state, but during peak and chronic EAE, they did observe the cells in the blood and the spinal cord. So again, kind of, deriving from the gut, and they end up at the brain. They do a couple of neat experiments looking at gut microbes. They develop a commensal ELISpot, which is cool. They took autologous. So when I say autologous, it's just from yourself. So fecal material from the same mouse. They heat killed it and put it on a ELISA plate, and then they came back and added the cells, and they could see, what antibody secreting cells were binding to the fecal material. And then you can also do so with that. They really wanted to understand like what proportion of cells were mostly binding to the commensals. And as we would expect, it's mostly IgA. I'm sure there were some IgG, but the largest proportion, when you look at the graph, was IgA. They do this really cool thing where they go into MS patients, and they're kind of digging into like this commensal question. And so they have a cohort of MS patients and healthy controls, and they quantified IgA-bound bacteria. They do this with something called IgA-seq and BugFlow, which is kind of a cool way to look at through flow cytometry to look at bacteria that has bound to IgA. And you can also do 16 siRNAs sequencing to look at the different types of bacteria. And so only in the MS patients that had relapsed had a significantly different trend. So they experienced lower percentages of IgA-bound gut bacteria in those patients compared to healthy controls. So suggesting that you are getting egress out of the gut in patients. You can't look at their brains, obviously they're living, but you can look at feces, you can look at IgA in the gut. And so I think that at least starts the question of, how is this relevant in humans?
Yeah, and they had originally shown that egress or that depletion of the IgA producing cells in the gut in the mice. And so this is the one kind of corollary, just as you said, that they can actually do in humans to show that this is relevant in humans.
Right.
And I think that there is something else here that might speak to that whole, like, it's really complex thing, because if certain IgA producers leave, and they leave those bacteria uncovered and able to expand, there are certain bacteria that produce metabolites that are anti-inflammatory, or certain metabolites that promote inflammation or promote immune responses or suppress immune responses. It's possible that there could be expansion of particular types of bacteria that then contribute back to either development or suppression of disease. in these
Yeah.
different situations. So it could be really
that's a good point.
complex.
Right. Like it's not just the IgA itself. It's what is it leaving as it, what is it leaving in the gut and how does the gut change?
Yeah, because
What
if
does
you
it
have
secrete?
expansion of certain gut microbes, there's sometimes that you can get expansion of beneficial microbes that will secrete more propionate or butyrate, and those are anti-inflammatory molecules that can then be circulating in the body and regulate immune responses. And we also know that some of the metabolites that are produced by the gut bacteria
Mhm.
can get to the brain and regulate brain function as well. And so, you wonder, could it be more complex than just the IgA producers that are making IL-10 in the brain? But in fact, some of the things that they're altering in the gut microbes, because they're producing less antibodies and the certain populations of microbes are allowed to expand in that situation, might also be contributing to the outcome of disease.
Right. Yeah, definitely. I would agree with that. And I think what would have been interesting too is to monitor the microbiome of the mice in each of these mouse models to try to understand maybe that better.
And I'm sure they have that, because if they did this bug flow, they can just sort those bacteria that are coated or uncoated, and then just see what kind of expansions or reductions in the
Sure.
community composition they get. And I'm sure they have that, and they can analyze those data.
I
Yeah,
feel
the next
I feel
steps.
I feel like we've planned out 5 more papers for them.
Exactly.
I know. This isn't, right. Like this isn't to criticize because there wasn't enough in this
No,
paper.
not at all.
No.
This paper
It's just
had
like,
enough, but
where could you go with this?
Definitely. And
It's interesting.
No, for sure. And it really does help kind of frame these next steps. And for a listener, like this is where our brains go as to, okay, this is the next experiment. You could do this, what you could do to prove this. And it's like your next Cell paper.
So make a big jump now. Let's assume this all is relevant to human MS, right? So
Right.
what's the problem in MS? Like, what are they not getting these neuroimmunosuppressive IL-10 producers in the brain? Do they not have them?
That's what I'm. Yeah. I think the answer to your question is there's not a lot of trafficking of these antibody secreting cells at steady state. So they're not there as they were there in the BAF overexpression model to just protect ahead of time. And so, your question of like, what could you do to help elevate? And that's kind of the last little section. So excellent transition into what they did was they wanted to find a commensal microbe that did just that. Could you elevate systemic levels of IgA to then help reduce severity? They used a protozoa, a commensal protozoa called Trichomonas musculus, TMU. And so in mice that were colonized with TMU, they exhibited less severe EAE, lower incidence of disease, reduced inflammation and demyelation, and higher IgA. And so it goes to show that, what happens in our gut could impact future disease. And so, like designer, commensal, looking at MS patients and the differences in their gut microbiomes, I guess what could be helpful.
So MS
Well, you
is
could
going to be
also-
a microbiome disease again, like everything else, right?
I know, that's why I'm like hesitant to, right?
Yeah, I mean, you could also
Yeah.
think about, are there ways you could deliver antigens to the gut in order to kind of induce the production of antibody producing cells in the gut to those antigens?
Right.
But could you just give people IL-10 who have MS? Would that help?
I
think that the problem there is the IL-10 won't make it into the brain.
Okay.
Because the blood-brain barrier and the blood-CSF barrier are pretty tight, and you have to give drugs to open the blood-brain barrier in order to get proteinaceous molecules into the brain.
although
we could
in
use
EAE, wouldn't it be already kind of open
MS.
or MS
Maybe.
rather?
Maybe.
Could use a viral
Just because-
vector that would
yeah.
get into the brain. Yeah, some AAV vectors get in and deliver the gene for IL-10.
IL-10. Yeah, that would be very cool.
That would
experiment.
work.
Hmm.
That would
Yeah.
work.
Well, maybe.
Maybe.
Well, you could try it. Let me put
Yeah,
it
you
that
could
way.
try it. Yeah.
So maybe in the patients. So I'm thinking about like rituximab that doesn't work for a proportion of patients.
hmm
Could you give them, if rituximab could make it, but I guess an antibody is bigger than IL-10. The IL-10 could possibly also make it. But then there's a problem with delivering like just a cytokine, right? Because there's all these things that cytokines do.
Yeah, sure.
And are you going to be more likely to be susceptible to viral infection because IL-10 is going to turn down your Th1? I don't know.
Tysabri, yeah. Back to Tysabri. That's right. Same.
Right.
Well, you have to balance it always, right? You have to do the trials and find out, but
one cool way would be to cage the IL-10 in some sort of nanoparticle that targets specifically to the blood-brain barrier, and they can release the IL-10 into the brain.
Yeah, that would be very cool. Right. Targeted approaches.
So in a recent-ish episode of TWIV, we talked about mechanisms that certain viruses use to get across the blood-brain barrier, and a new mechanism that West Nile could use. And so maybe we could look at some of those mechanisms of crossing the blood-brain barrier and try to harness them.
Do you know if lipid nanoparticles get into the brain? Do we know that?
We need a neuroimmunologist to come on.
You need to be able to get those lipid nanoparticles to access the brain in the first place. I'm not sure how many situations we've had where we put the lipid nanoparticles, say, in the blood, so that they even could get to the brain to try to cross the blood-brain barrier.
Right.
You could, yeah, I don't know.
Because if they did get in, then you could put an mRNA in them for IL-10.
Yes.
mRNA will
inject
cure.
it into the CSF, but
Mm-hmm.
I don't know. That's kind of
That is tricky. Yeah.
invasive.
Right. But
They do inject things into the ventricles. I mean, it's not something you want to do on a regular basis, but
right.
Well, if you have MS,
do that.
if you have MS and it's a big problem, you can do things like that, right?
Yeah. So I'm thinking, because if this is going to be targeting the percentage of people that are not responsive to rituximab, I don't know how, 20%, then
They're going to want something.
So I
don't know. I assume you could go, through, I mean, it's not easy. You have to drill, right? But you could inject into the ventricles and put IL-10 in the ventricles. I don't know how easily it would get around to the areas of the brain where it would need to, or whether it would be flushed out or not. I don't know.
Yes.
You could put the adenoviral producing IL-10 virus into the ventricles, and then maybe that would work. I don't know.
Yeah. But I think that what this paper, at least for me, was successfully able to do is to prove to me that these cells do end up in the brain. They do, make a difference for this disease, and that it isn't the antibodies themselves. It's IL-10 and potentially, other things that, IL-35, they didn't really look at. They did mention in the paper, but didn't look at. Right. So yeah, what do we all think? Any last thoughts?
Yeah, I mean, to me, I was just really excited, and maybe this is because I've been thinking about teaching these topics to undergrads, and so I'm simplifying things in my brain. But to me, I was like really liking the focus on plasma cells doing something other than antibody production.
Yes,
and sort of the focus on that. And also, this really made me think a lot about compartmentalization in the immune system and the way we think about the mucosal immune system, and whether or not how that is or is not true, and how the brain fits into that compartmentalization.
Yeah, because I think that, how I've thought about it for sure is that we talked about licensing them, right? We talk about homing back. So if you're educated in the gut, you home back to the gut, right? And so this is saying that doesn't necessarily hold true, that you can leave the gut, but we always think about those retaining chemokines and integrins that maintain you in your location.
Right.
But that might not entirely hold. And so this generalized inflammatory response might then lead to mobilization of these. But then you wonder what else is being mobilized. Like, are you just moving your B cells around all the time from your gut whenever you get some sort of infection anywhere? I don't know.
Yeah. And that's why I was really curious about where can they go? Can they only go to the brain, or could they go to other places?
Yeah. And one of the things in this paper, I think they had talked about, okay, so there's I think threshold was what they use in the firewall, that there's a firewall that constrains IgA responses to mostly commensal bacteria in the gut. And they're not really going other places. But studying pregnancy, in multiple different animal models, you can detect huge amounts of IgA and biosecreting cells to rotavirus, to flu in milk. And so I don't know, does pregnancy breach that firewall? Is that not considered a homeostatic state because now you have a developing mammary gland that's secreting the things that cells need to traffic there, CCL28. But we find at a steady state during pregnancy, she's an animal or a woman is not in some, she doesn't have preeclampsia. We find these cells. So I think it asks the question of like, what is homeostasis, and is that firewall? I don't know. It's kind of squishy.
And that reminds me that a lot of the autoimmune diseases tend to be X-linked, right? I don't think this particular one is, although I'm not entirely sure. But for those that are, there's probably a sex bias too.
Right. And one of the reasons why I've been thinking a lot about this topic is I'm working with some pill-based vaccines where it's an Ad5 based vaccine. It releases the antigen in the gut. And, can we get IgA and biosecreting cells to the sites of respiratory infections? Mm-hmm. You can imagine which respiratory infections we're interested in. And
I have no idea.
Anyone. But so if they migrate to the nose, are you kind of helping to block that first site? And how well can you get that stimulation? And does it depend on other, right? What's that firewall consist of?
All right.
Stain your cells to see if they're
great.
Making IL-10 also.
I know. I think that is going to be the next thing I do. I just have to find an anti-ferret and anti-hamster IL-10, which let me tell you that finding ferret and hamster reagents right now is a bit of a challenge. So
I imagine so.
All right, let's wrap it up.
What do we think? Yes, go ahead.
You have to go, so it's time to wrap up, right? 4:20?
Yeah, 4:20. I do have a meeting at 4:30.
Yeah, we have some business to take care
We
of.
We do.
So we'll have to
get we're getting a regular podcast on the schedule for Vincent's sanity.
All of our sanity.
I think it helps
sanity,
you guys
yes.
too. Yeah, don't do it just for me.
Sure.
All right, that's Immune 44, microbe.tv/immune. For the show notes, send your questions and comments. We got a lot of them backed up. We'll get to them.
Yeah.
Immune at microbe.tv.
Next show, maybe.
Yeah. And if you like what we do, support us. Microbe.tv/contribute. Cindy Leifer's at Cornell University. Cindy Leifer on Twitter. Thanks, Cindy.
Thanks. That was a fun discussion.
Steph Langle's at Duke University where there's no gas. Stephanie Langle on Twitter. Thanks, Steph.
Thank you. This was so much fun.
Brianne Barker's at Drew University. BioProf Barker on Twitter. Thanks, Brianne.
Thanks. It was great to be here. This was really fun to come up with ideas.
I'm Vincent Racaniello. You can find me at virology.blog. Music on Immune is by Steve Neal. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.